Okay. Yeah. Hi, everyone, and welcome to this problem solving session for experimental biotechnology. We'll be discussing the content from week 5 today. My name is Nandika. I'm a PhD student from IIC Bangalore. And yeah, let's get started. As always, if you have any questions, feel free to drop them in the chat or just unmute yourself and you can ask me. So, what we learned in the last week was a technique called size exclusion chromatography, which is also called gel filtration or gel permeation chromatography. And it is basically a technique that allows us to separate molecules based on their size. Um, I mean, we always say it's based on their size, but it's actually not directly the molecular weight, but actually more related to the shape of the molecule. Okay, and that's something we'll discuss further along in this session. So it's actually separated on the basis of something called the hydrodynamic radius. And this is done by passing the molecule through a solid phase which is porous, which is basically acting like a sieve. Okay, it's acting like a sieve to separate the molecules on the basis of their size. And that's because it has pores and is usually made up of cross-linked polymers. So the solid phase will be made up of beads like this. Okay. Yeah. Solid phase will be made up of beads like this. And if we zoom in on the bead, then we can see all the pores which are present over here. Right? And when we have molecules of different sizes, not all of them will be able to enter this pore. So if we say the pore has a profile like this, okay, it has a decreasing diameter along as we go inside. Then if I have a very small molecule, it will be able to go all the way inside the pore and then come out. But if I have molecules which are slightly larger, then they'll get stopped somewhere here, right? So here the diameter of the molecule is bigger than the diameter of the pore over here. So it won't be able to travel as far as this brown molecule. And the green molecule can just like maybe barely enter the pore and then come out. So if we look at it in this way, then the distance traveled by this brown molecule, which has to go all the way inside the pore and come out, is going to be much more than the distance traveled by this green molecule, right? So if we put this mixture of molecules into our mobile phase and we just pass it through our column, what is going to happen is because the big molecules have to travel less distance, they're going to come out faster, right? So as this moves through the column, the big molecules have to travel less distance, they will come out first. And the small molecules, because they are traveling through all the pores, they're traveling a longer distance, and so they get separated from the bigger molecules and will take more time to elute from the column. Okay, so the profile that we get would look something like this, where the largest molecules come first, then medium sized molecules, and then the smallest molecules. So, how can we get this shape like this? What happens is that when the mobile phase is eluting from the column, we can add a detector over here. Okay, so that might be, say, a UV detector. So just detect UV absorbance at maybe 280 nanometers. Since we know that proteins have amino acids like tryptophan and tyrosine, those all absorb light at 280 nanometers. So what the absorbance profile will basically give us a peak of all the proteins that are coming out of the column. Okay. So couple of concepts related to size exclusion chromatography, or specifically some terms that are used quite often. obviously there is the total volume of the column. Okay, so the full column volume is just the entire volume which is occupied like this. But you can see that all these spheres are part of the solid phase, but it is not completely filling the space in the column, right? All the space is still empty, right? You even have this space between these spheres, space between the beads, which is empty, right? So this is not part of the solid phase itself. This is part of the mobile phase. And this is something called the void volume, right? So all this portion in blue, which is excluded from the beads, this is called the void volume. Okay? And so if the total volume of the column is made up of the void volume plus the volume of the bead plus the volume of the pores inside the bead, right? Now what happens is this all our proteins are going to pass through the pores, but the our elution is going to pass through the void volume first. So until the void volume passes through the column, none of our proteins can elute. Okay, all our proteins will elute after the void volume. The void, since our stationary phase is made up of spheres, you might have learnt in your previous classes about packing efficiency of spheres and so on. And so, let me see. If I have spheres and I pack them, they will fill up roughly 66% of the space, okay? Which means that 33% is empty. And this 33% is the void volume. So if I have a column volume of 24 ml, that means that 8 ml will be the void volume of that column. Okay? And this is true for both pre-packed columns and columns that you pack yourself in the lab. Okay? Another concept that you may have seen a lot is something called the distribution coefficient, or KD, where it basically describes how much time, or how much, it's basically a function of how much time your protein is spending in the pore, right? So if I have some protein which is eluting after some time from this column, I'm getting some elution volume for that protein. So the KD is defined as elution volume minus the void volume on top of the pore volume. Okay? KD has to be between 0 and 1. So if KD is 0, then the sample is not going inside the pore at all, right? So that means that VE is equal to V0, right? The sample is not entering the pore at all, it is directly coming out in the void volume. When KD is 1, that means that this KD minus V0 is equal to the volume of the pore, right? So that means that the sample is fully entering the pore and coming out. So it is the smallest possible molecule that can come out of the column. If I have KD which is greater than 1, okay, this should not be possible in size exclusion chromatography. KD greater than 1 means there's some other kind of interaction which is going on between your protein and your column. So maybe your protein is getting adsorbed on the column, or maybe it's interacting with the solid phase or in some other way. That is the only way in which KD can be greater than 1. Okay. Now, another thing we talked about is that we're separating it basically based on the shape of the molecule. And now, obviously, the shape of the molecule is proportional to the size, right? So if I have two proteins A and B, and A has molecular weight of 10 kDa and B has molecular weight of 100 kDa, then obviously B is going to be bigger than A, right? So B is obviously going to be bigger than A. So obviously this the size of the molecule is proportional to the molecular weight. But so far we have discussed proteins which are all round, right? Or what we call globular proteins. Not all proteins will necessarily become spherical, right? So obviously you can have globular proteins which behave like a sphere, and they will follow all these equations happily. But what if my protein is folding into some shape like this, right? These two may have the same molecular weight, but obviously they are a different shape. Okay, so they will pass through the size exclusion column in different ways. So again, let us consider this is one bead from a stationary phase, and this is the pore. And I have two molecules. One is this sphere, one is this rod. Now this sphere can easily go inside, right? So it can go like this and come out. But if I have a rod of the same molecular weight, then there are two possibilities, right? The one possibility is that the rod won't be able to enter at all, right? If the rod comes in this direction, won't be able to enter the pore at all. The other possibility is that the rod can come like this, right? And it'll get stuck somewhere else. So obviously, the rod is kind of behaving like a bigger molecule compared to the sphere. So if both of these elute from the size exclusion column, then what I might expect is that my spherical protein might elute somewhere here, but because my rod is acting like a bigger molecule, maybe it will elute faster from the column. Okay, just because of its shape. So today we'll discuss mainly globular proteins because that is what most people deal with in the lab. But do keep in mind that you can have proteins which are different shapes, which will affect the way that they behave in a size exclusion column. Okay. If anyone here is working with membrane proteins, what we generally do for membrane proteins is we have a protein like this. But because it's hydrophobic on all the sides, this region is hydrophobic, you have to keep it in a detergent micelle. Okay. So you can see immediately that this entire structure is going to be elongated. It's not going to behave like a sphere. Okay. So these are just things to keep in mind if you're working with some unusual proteins. But anyway, so this is what a typical size exclusion profile might look like. Here we're going to add a sample. Remember, we have the void volume, which is the region between all the stationary phase beads. So a sample cannot elute before the void volume. So the void volume is ending over here. And now the high molecular weight samples will elute first because they're going to take a shorter path through the column. Then the intermediate molecular weight, and then the low molecular weight. Right? And this is the total volume of the column, and everything is eluted within one column volume. Okay. So this is just theoretically what a size exclusion chromatogram will look like. Now we can obviously use size exclusion chromatography to separate different proteins of different sizes. We can also use it to separate protein from salts. So this is something called desalting, where if you have a very small pore size, then all your proteins are going to go out, right? All the proteins are going to go around the bead. They can't enter this pore. But all the ions and salts and so on will have to pass through these pores. And so what you collect here just after the void volume will be all the proteins, whereas the salts will be separated from that. Okay. So this is another application of gel filtration or size exclusion chromatography. It's called desalting. Right? Makes sense. If I make the pore size small enough, I should be able to separate my proteins from my salts. So this can be very useful if you have a very high concentration of some salt in your buffer and you want to get rid of it. Or maybe you want to change the buffer in your sample or anything like that. You can use this protocol. And we just saw what an ideal chromatogram will look like, right? You want peaks like this, which are well separated from each other. Peaks are nice and symmetrical, and they're all eluting between the void volume and the total volume, right? So this is ideally how our separation should take place. But maybe for some reason our peaks are coming very close together. So here it's difficult to separate these two peaks, right? Because if I extend this peak, it will come down like this. And this peak will come down like this. So this region in the middle is where these two proteins are going to mix, right? So that means that these two proteins have not been separated properly in this column. So that could be due to a number of reasons. It could just be that the column is not appropriate for our application. Could be due to the flow rate, could be due to sample preparation and so on. So there are a number of things to optimize if you see something like this. Now, another issue you can have is that instead of getting a nice symmetrical peak like this, the peak starts to become larger on one side. So if it's larger, if this side of the peak becomes larger, it's called tailing. And if the peak becomes larger on this side, it's called fronting. And both of these are issues that occur due to improper packing of the column. Okay? Column is not packed properly, you can get issues like this. Another issue you can get is that this is my void volume, and ideally I should get peaks starting from here, right? But instead I'm getting some peak like this before the void volume. So this is also an issue with column packing. What is happening? What is happening is that instead of a well-packed bed like this, okay, there is maybe some gap in the bed. So instead of going like this, our protein is going directly like this through the bed. Okay? So this is called channeling. This is something called channeling. And again, all of these peak issues are caused by packing, improper packing of the column. Okay? So if we start seeing things like this, then maybe you should take out your resin and pack your column again. Now, this is another scenario that we discussed just now, that what if my peak is coming after the total volume, right? If my peak is coming after, so here is where my elution should have stopped, but I'm still getting peaks over here, right? So this means that this sample has KD greater than 1, okay, which means that our sample is interacting with our resin, our sample is interacting with our beads. So maybe we need to change the buffer composition or change the kind of resin that we're using if this is the case. Okay. I mean, obviously you can understand that if we are now including interactions with you can understand that if our sample starts interacting with our resin in ways, then the separation is not going to be based purely on size, right? It's also going to be based somewhat on the affinity for the resin, right? So then we cannot use this as an accurate measure of the size of the protein, right? So ideally it should not interact with the resin at all. Now, columns have some terms like exclusion limit and penetration limit. Okay, and these are both related to the pore size of the column. Okay, again we'll go back to this diagram. Okay, and so this is my resin, this is one bead from the resin, and this is the pore. And so this distance, any molecule which is bigger than this will not be able to enter the pore at all, right? So it will also come out in the void volume, right? If my molecule is this big, it won't be able to enter at all. So basically, this distance is the exclusion limit, beyond which the molecule cannot be separated by this column, right? Because this is my exclusion limits. Now it doesn't matter if my molecule is this big or my molecule is this big, right? These cannot be separated anymore by this column. They're both going to be excluded from the pores and they're going to elute together, right? So therefore, the molecule cannot separate any further than this. The penetration limit is telling you the last, like basically the lowest molecular weight until which column the samples can be resolved. So this is basically related to this diameter. Okay. So again, let's consider the pore. So there is some minimum diameter of the pore like this, right? So now if I have some molecule which is this size, it is going to go all the way inside the pore and then come out. But if I have another molecule which is this size, right, it can also go all the way inside and come out. So when we look at the separation of the column, these two are both going to elute together at the end because the column cannot differentiate between these two proteins. Okay. So this basically smallest diameter of the pore is called the penetration limit. Okay. So for example, for this column, what they're saying is penetration limit is 1 kilodalton. Okay. Any protein which is less than 1 kilodalton cannot be separated in this column. Okay, it is all going to elute together in this same peak. And here they're saying exclusion limit is 3.2 into 10 raised to 6, which is basically 320 kilodaltons. They're saying that everything which is bigger than 320 kilodalton will come together in this peak. We cannot separate them if they're bigger than 320 kilodalton. So any protein which has a size between these two limits can be separated by the column. So for example, this is saying this is a protein, right? This is some protein of intermediate size which can be separated well in this column. If I had some smaller protein, okay, maybe this is 5 into 10 raised to 4 Dalton. Okay. So both of these can be separated well in this column because they are lying between the exclusion limit and the penetration limit. So what is the matrix of these columns made up of? So like we discussed, it's made up of cross-linked polymers. So you can see here, this is agarose and dextran cross-linked. Okay. And the nature, like the ratios of the two polymers, how much of them is added and so on, will affect the pore size of the matrix. Okay. So this cross-linked dextran and agarose is a matrix called superdex. Okay. And within superdex, you can see there are different varieties. Superdex 30, Superdex 75, Superdex 200. And you can see that this is what this is saying is the fractionation range. So basically, what is this? Right? What is the range between here and here? This is saying that the fractionation range for S30, Superdex 30 is like 100 Dalton to almost 10 kilodalton, right? S75 basically is telling you it has an exclusion limit of 75 kilodalton. S200 has a higher exclusion limit. So you can see that these have different fractionation ranges. So depending on the proteins that you want to separate, you have to pick what is the right matrix for your application. So if I have a sample with very small proteins, then maybe I should be taking this. But if I have some very large protein, okay, I have some protein which is, let's say, 200 kilodalton. If I run it in this column or in this column, it will come out, it is beyond the exclusion limit. Okay, so it will come out in the void volume. So if I have such a big protein, then obviously I have to go for a matrix like this, right, where I can safely run my 200 kilodalton protein, and it will come within the fractionation range. Obviously, superdex is only one kind of resin. There are many kinds of resins depending on what polymers they're made of. So if it's just dextrans, then it's called cephadex. And obviously, again, depending on the ratio and the concentration of the dextran, you can have different fractionation ranges. Right? Cross-linked dextran is called cephacryl. If it is only agarose, then it's called cepharose. You can also make a polyacrylamide size exclusion column, right? And anything with pores. So you can have polyacrylamide columns, and they are called biogel. Okay. So obviously you have different substrates with different fractionation ranges. And just an example of what happens when you run the same protein in two different columns. Okay, so this is actually my data. So this is the same protein, but here I've run it in a column called superose 6 increase. And here you can see that the fractionation range goes up to like 5 into 10 raised to 6, right? Which is 5000 kDa. And here I've run the same protein in another column called superdex 200, which has a smaller fractionation range. So this is a fractionation range up to about 600 kilodalton. Okay. And what you can see is that because my protein is kind of in the middle of the fractionation range of this column, it is going to elute at 15 ml. But because my protein is more towards the beginning of the fractionation range of this column, it is going to elute much earlier. So the same protein is eluting at 10.6 ml. Just because the pore size of the column is different. And so because this column has a smaller pore size, my protein will elute earlier from this column compared to this. Okay. So again, depending on the size of your protein, you have to pick the column which is right for your application. Now, obviously, we know that the size and the molecular weight of the protein are related. So if you want to determine the size of your protein from SSE, you can run something called a standard. Okay. So this will give you something like this, which is called a calibration curve. So a standard is just basically you will get a mixture of known proteins, and when you run it on the column, you should get the same number of peaks, and you already know what the weight of the protein is. So how the calibration curves works is that we know that log of the molecular weight is proportional to elution volume. Okay. So all we have to do to make the calibration curve is plot log mol weight versus elution volume, and we should get a straight line, right? If our column is working properly and there are no issues with packing and so on, then we should get a straight line, right? And we can use this line to basically find the molecular weight of any protein that we run in the column. So I'm running some new protein. I don't know anything about it, but I'm getting a peak, right? So I'm getting a peak somewhere here, right? Then I can just use this curve to find what the approximate molecular weight of that protein could be. Okay. So maybe you can see it clearer here. So this standard is basically a mixture of these 5 proteins, and when I run it in the column, I get these 5 peaks. And obviously, I'll assume that these peaks are there in decreasing order of molecular weight. So then I can assign the peak to each protein and make a graph like this. Okay, so let's see how to make a standard graph. Okay. So here, when you run a size exclusion chromatography experiment, you'll get a lot of data out of it. Okay, and it might look something like this. So especially if you use a system such as the ActaPure or any computerized system, you'll get a lot of data which might look something like this. But just to take you through what data you can get, so this is, here you'll see UV, right? This is telling me that in my UV detector, after this many ml of elution, this is the value of my UV absorbance. Okay, so this is basically the trace which is telling me how my protein is eluting with time. Now, you can also have other detectors. So we also have a conductivity detector. So this is just going to tell me when the salt is going to elute. Okay, but this is the standard run that I ran in one of the columns. So let's try and see if we can plot this data and make a standard calibration curve. So for my standard data, I just need to plot these 2 columns. So I'll just copy these 2. Okay. Get rid of the header. Okay. And I just need to plot this as an XY plot. Okay. Okay. So here, if you see, I now have a chromatogram, right? Where this right, I'll just add some titles so it's clearer to everyone. So this is elution volume. In ml. Okay. I hope this looks familiar to all of you as a chromatogram. Okay. Now, we already know that the void volume, nothing should be eluting before the void volume of my protein. But I can see that my total column volume is around 30 ml. So my void volume should be 10 ml. So I will disregard these 2 peaks. Okay. Maybe there was something stuck in my column or whatever, but these peaks will not correspond to my protein. So we'll consider these 5 peaks over here. Okay. For plotting our standard. Now we need to know which proteins were there in our standard. So when you run your standard, you will get a small booklet along with it which will tell you which proteins are there. So I found this manual for my gel filtration standard. Okay, so this is a gel filtration standard from Biorad, where they will give you a lot of instructions on how to use it, etc. But what we're interested is in this part, okay, which is telling you the components. So here is telling me the component and the molecular weight. Okay, so here I can see that I have 5 proteins in this compound of molecular weight 670 kDa, 158 kDa, 44 kDa, 17 kDa, and 1.3 kDa. And these will correspond to my 5 peaks over here. Right? So now what I have to do is find the elution volume of these 5 peaks and then plot that versus the log of the molecular weight. Okay. So let me just list the elution volumes of these 5 peaks. So this has elution volume of 12.5. This is 16.0. This is 20. Okay. This is just an approximate estimate, but this will be maybe around 23. Yeah, okay, 23.1. And this will be somewhere around 28 ml, I think. Right? 28.8. Okay. 28.8. So these are the 5 peaks that we got in our standard run. Okay. Now we can match them to the molecular weight. So we had 670 kDa, 158 kDa, 44 kDa, 17.5, 17 kDa, and 1.3 kDa. Okay. This is just the molecular weight. What we need to find is the log of the molecular weight. Okay. So just quickly find that. Okay. So now this is what we need to plot to make the standard curve. Okay. So we'll just make this as a new plot. Okay. Already you can see that this is kind of formed a straight line, but let's just quickly add the trend line and okay. So here we've got the equation of the line, and we've got a very high R squared value, right? So that R square is basically measuring how straight our line is. So R square can be maximum 1, and we have R square of 0.995, okay, which is basically telling us that a standard run is probably very accurate, or very precise rather. And what this is telling me is this is an equation that I can use to find the molecular weight of any unknown protein that I will run in this column now. Okay. So now if I have some unknown protein, okay, so I have some unknown protein and I am getting some elution volume of say 18 ml. Okay, I want to know what could be the size of this protein. So what I just need to do is put it in this. I just need to put it in this equation, which is the equation of this linear trend line. Okay, so 18 is equal to minus 6.1712x plus 48.503. So x will be equal to 18 minus 48.503. Right? And then we'll have to divide by this. .1712. Right? So now what I've gotten here is the value of log of the molecular weight. So what I have to do is take the anti-log of this. okay, which is basically 10 raised to this answer. And what this has given me is this number, which is about 87.65 kDa. Okay, so if my protein is eluting around 18 ml, that means it has approximate molecular weight of around 88 kilodalton. Okay, and this is a very powerful tool to estimate the size of your protein, right? Because you only have to do one standard run for your column, and then for when you run any new protein in your column, then you can easily estimate what its size could be. Okay, just one word of caution though regarding these standards. Obviously, this standard run has worked very well for this column, but you also have to keep in mind what kind of column you are using. Okay, so this is the same standard run, okay, but this is run in an S75 column, okay, which means that the exclusion limit of this column is 75 kilodalton. Okay, but if you remember, we have components of our standard which are more than 75 kilodalton, right? So these two, like 670 kDa, 158 kDa, these will not be separated by the column at all. So therefore, we will not see these peaks in our standard run. Okay, so if we try to plot this data, okay, you'll see I'm only getting 4 peaks, right? I should be getting more than 4 peaks because I have 5 proteins. But because the exclusion limit of this column is 70 kilodalton, the first 2 peaks are coming together. So both of the first 2 proteins are coming together in this peak, which is also why this peak is much higher than the other peaks. Okay, so this is the first 2 proteins. This has 44 kDa. This is the 17 kDa, and this is the 1.3 kDa protein. Okay, so I cannot consider this point at all in my standard analysis. Why? Because all of these proteins have been excluded by the column. So this is not an accurate representation of the size of the protein. So if I want to make a standard curve for this column, I should only use these 3. Okay, so let's do that. So I have 44 kDa, 17 kDa, and 1 kDa. Okay, so this is around 10. This is 12.5. This is somewhere closer to 18, 18.2. Okay, make log of the mole weight. Okay, and now I can make a standard curve for this column, but it is a standard curve with only 3 points. Right? So this may not be the most trustworthy standard curve, but we are able to fit this standard curve for this column. Okay. Another thing I want you to note that obviously since this column has a smaller exclusion range or like a lower fractionation limit, notice the separation between the 17 kilodalton protein and the 1 kilodalton protein, right? So these two have been separated very well in this column. Whereas if you look at the previous column, these 2 peaks are much closer together, right? So obviously this column is better for proteins with large molecular size, but this column is probably better for separating proteins of low molecular weight. Okay? But yeah, anyway, so this is basically how you can create a standard curve or calibration curve for any column. Obviously, you will have to run the calibration curve separately for each column because I cannot use this calibration curve for my other column. Okay, this is unique for this column. So you have to keep that in mind when you do experiments like this. Yeah. So let's try to look at a problem related to this. Here they're saying estimation of relative molecular mass. They're saying relative molecular mass is investigated by exclusion chromatography using some S300 column. They have this mixture of proteins as a standard run, and they've obtained this elution data. What they want to know is what is the approximate size of the unknown protein. Okay, so we just discussed how to do this, so let's solve this problem. Okay, let's make a new limit. So our elution 22.5, and this has a molecular weight of 158 kDa. Okay, 21.4, 210 kDa. 18.2 and 444 kDa. 16.4 and 669 kDa. 13.6, and this is a blue dextran, which is much bigger. So this is saying 2000 kDa. Okay, and we have some unknown protein which is eluting at 19.5. Okay. So just looking at the elution volume, we will expect it to sit somewhere here. So between 444 kDa and 210 kDa. So maybe this protein will be somewhere around 300 kDa, right? But let's do a proper estimation and find out. So obviously, step 1 is to take the log of this to plot our standard curve. Okay, this will take our elution volume here, and we'll plot our standard curve here. Okay, let's add a trend line. Okay. So here we've gotten the equation of our standard curve. Okay, and we can use this equation to find the approximate molecular weight of our unknown protein, like we just did earlier. Okay, so 19.5 is y. Right, so 19.5 will go somewhere here, it will come down here. But let's just solve the equation and find out what is the value of y. Right, so y will be equal to 19.5 minus 65.074. This divided by minus 8.2. Okay, so this is giving me log molecular weight of my unknown protein. To find the actual molecular weight, I have to take the antilog of this, which is giving me an approximate molecular weight of 337 kilodalton. Okay, right? Makes sense. So even visually we could tell that it was around 300 kilodalton, but by using the calibration curve, we've gotten a more accurate value of 337 kDa. Okay. So approximate MR will be around 330 kDa. Okay. We can do some more problems. So here they're saying if we have a mixture of molecules, so it's containing hemoglobin, myoglobin, and myosin. Hemoglobin is 65 kDa, myoglobin is 17 kDa, and myosin is 180 kDa. What order will they pass through a size exclusion column? So as we've just discussed, the bigger molecule will elute first and the smaller molecules will elute later. So obviously the biggest molecule will be the first to appear. So that is myosin, right? Because it is 180 kDa. The second molecule will be hemoglobin, which is 65 kDa. And the third molecule will be myoglobin, right, which is 17 kilodaltons, right? Just in decreasing order of size. If the size exclusion chromatography column is said to have an exclusion limit of 40 kilodaltons, will hemoglobin, which is 60 to 65 kilodaltons, be fractionated or excluded from the column? Now remember, the exclusion limit is the largest possible molecule which can be correctly resolved by the column. And if our protein is larger than the exclusion limit, then it cannot be resolved by the column. Okay, it will be excluded because 60 kDa is greater than the exclusion limit. Therefore, it will be excluded from the column. Right? It will not be able to enter the pore at all, and it will be excluded. Would vitamin B12, which is 1.3 kDa, be fractionated or excluded from the column? Again, the exclusion limit is just saying that anything bigger than this will be excluded from the column. Obviously, 1.3 kDa is much smaller than 40 kDa, and so vitamin B12 will be fractionated by this column. Okay. Okay. We have a globular protein of size 30 kilodaltons. It exists as a monomer and a dimer. What would be the most appropriate technique for separating these two proteins? Or rather, these two forms of the same protein. Since these are two forms of the same protein, most of the properties of the protein are going to be the same, right? So things like the surface charge or the hydrophobicity and so on of these two forms will be pretty similar. So the most obvious difference between the monomer and the dimer will be the size. And so the easiest way to resolve these two will be through size exclusion chromatography, right? Because we can easily get two peaks. This will be 60 as the dimer. This will be the monomer, and we can resolve the monomer and the dimer easily. In size exclusion chromatography, molecules are separated based on their dash. Okay. Obviously, it is not based on charge or adsorptive properties, but remember what we said, they are separated on the basis of their shape, right? Not necessarily on the molecular weight, it's more based on their shape. So here the answer will be hydrodynamic volume, right? Because as we discussed, even if you have the same molecular weight but a different shape, you will move, the two proteins will move differently in the size exclusion column. So the answer for this will be hydrodynamic volume. They're asking, effective separation of the protein sample through the SSC column depends on which of these factors? Pore size, obviously. So the pore size should be appropriate for the protein sample. If the pore size is smaller than your protein sample, it's not going to get separated at all. If the pore size is much bigger compared to my protein, then again it's not going to get separated properly. So pore size, obviously. Viscosity of sample and buffer, yes and no. This will be a very minor factor, but obviously the buffer should be such that the sample is not interacting with the column, and our sample should be stable in the buffer. Packing density of the column. Yes, obviously the column should be packed properly, okay, so that we don't get channeling effects and so on. So let's say that the answer for this is all of the above, because all of these are important for effective separation of the protein sample through SEC. Here it's asking, what do you mean by resolution in size exclusion chromatography? Is it the degree of separation between the peaks? Is it the range of separation of the column? Or is it the efficiency of the column media to produce a sharp and narrow peak? Okay. The answer for this is actually both A and C. Okay. Because degree of separation is directly related to how sharp your peak is. Okay. Imagine I have a chromatogram like this and I'm getting two peaks like this. Okay. Since these two peaks are very sharp, they are well separated from each other. Okay, so these samples are not mixed at all. So this protein is completely kept in this peak. This protein is completely kept in the other peak. But suppose my column media is not an efficient column media and it is not producing a sharp peak. Okay, instead I'm getting a broad peak. So I can still get one peak over here and one peak over here. But because I'm not getting a sharp peak, this peak will come like this. This peak will come like this. Right? Now, even though the top of the peak is in the same place, you would not say that these two proteins are separated from each other properly, right? Because there's such a big region of overlap between these two peaks where the two proteins are mixed. So even if I try to pick up this sample over here, I'm getting a little bit of this other protein in it also. Right? So resolution depends on the degree of separation as well as the sharpness of the peak. Yeah. In size exclusion chromatography, proteins are detected by measuring their absorbance at dash. So in SEC, like in other forms of chromatography, you can have different detectors, many different detectors. But I would say that the most common one will be detecting absorbance at 280 nanometers. Okay. And that is because the aromatic amino acids in our proteins, mainly tryptophan and tyrosine, and there's a small contribution from phenylalanine, absorb mostly at 280 nanometers. Okay, so this is another thing to keep in mind. So sometimes, in a very rare case, you might be working with a protein which doesn't have any tryptophans. Okay, so then you have to keep in mind that you might get a very low signal in your absorbance detector. Okay, but in general, in SEC, we will detect the protein by measuring absorbance at 280 nanometers. Okay. Application of SEC are isolate one or more components from a protein mixture. Yes, obviously. Determine molecular weight of the protein. Yes, again, as we saw that if you have a standard curve, you can determine the molecular weight of any unknown protein. Buffer exchange and desalting. Yes, like we discussed in the beginning, if you have a column with a very small pore size, then you can use it for the process of desalting. So application of size exclusion chromatography is all of the above. Okay. Chromatographic media used for SEC are chosen on the basis of dash. Inertness. Yes, obviously we don't want the resin itself to interact with our protein, so it should be inert. It should not react under different buffer conditions. If I'm running it at my protein at very low pH, the SEC column should still work as an SEC column. So it should be inert and it should be chemical and physically stable. Right? So both A and B will be the correct option for this answer. Okay. Molecules that do not enter the chromatography matrix are eluted when? So this is basically saying molecules which are not able to enter the pores. Right? So these are molecules which are excluded from the pores. When will they elute in this SEC run? Okay. So like we discussed, columns will elute between the void volume and the total volume. Molecules that do not enter the chromatography matrix will be the first molecules to elute. Right? They are traveling the shortest path in the column. So they will elute here at the void volume. This will be everything which is bigger than the exclusion limit of our column. It will not be after 1 CV, right? After 1 CV, we should not be getting any elution. And when half column volume of buffer has passed, that is already beyond the void volume, right? Remember, void volume is about 1/3 of VT. Okay, so these molecules are entered in the void volume. Fractionation range of a column matrix means dash. Is it range of molecular weights that have partial access to the pores of the particle and can be separated? Yes, this seems correct. What are the other options? Size of the molecules that are excluded from the pores. No. Right? Size of the molecules that are excluded from the pores is the exclusion limit. Molecules that elute in the void volume, again, this is exclusion limit. Is it selectivity of the matrix? No. So the fractionation range means the range of molecular weights that can partially access the pores. It is basically the range of weights that can be separated by the column. What might be the possible reasons behind a blocked column? Sometimes when you're running SEC, you see that the pressure is going extremely high. Okay, that means your buffer is just not passing from the column. So what might be the reasons behind this? Is it protein precipitation in the column? Yes, obviously that can block your column. Okay? So the protein has come out of the mobile phase, okay, just precipitated as a solid in your column. Column is not equilibrated properly. Uh, this will usually not lead to a blocked column. If this is the case, then maybe the SEC run will not separate the proteins properly, but it probably won't block your column. Column is overloaded with sample. Yes, there is too much sample. Okay, it is, so even the capacity of the column is, it has gone beyond the capacity of the column. So both of these can block your column. Okay. Both A and C. Okay. Next set of questions. Another term for size exclusion chromatography is gel filtration chromatography. Okay. So gel filtration or gel permeation. Desalting is a very specific application of SEC. Okay. So any general SEC run is not called desalting. Desalting is specifically with a very low pore size matrix. So, but a general term for SEC is gel filtration or even gel permeation. Gel permeation chromatography. Okay. What are the important factors to be considered while preparing a buffer for SEC? Okay. Buffer must be compatible with protein stability and activity. Yes, obviously. Right? We are doing SEC because we want to use our protein later. So we don't want our protein to be denatured in SEC at all. Buffer must be degassed, buffer must be filtered. Okay, these are two very important steps you must do before injecting buffer into a column, because you don't want to have bubbles which come into your column. Okay, if your bubble enters the column, it can destroy the packing of the column. So all three of these are very necessary before we use a buffer for SEC. Okay. It is necessary to degas the buffer, it is necessary to filter the buffer, and the buffer that we choose should be compatible with our protein. Okay, we have 4 proteins with different size, which will elute second in SEC. So let's just try to assign the elution order. Obviously, they will elute in decreasing order of the molecular weight. So starting from the biggest, so this will be the first, 145 kilodalton. RNA polymerase will be the second. Ribonuclease and cytochrome have very similar molecular weights, but we will assume that this column is able to separate them properly. So ribonuclease, which is 13.7, will elute first, and cytochrome will elute last. So they're asking which will elute second, and that will be RNA polymerase from this mixture. Superdex media is made by Dash. So, as we discussed earlier, Superdex, this is just a technical question. Superdex is made by cross linking dextran and agarose. It's the brand name Superdex. Ah, Analytical size exclusion chromatography is used for Dash. Desalting, checking the quality of the sample, or isolating one or more components from a sample. And all of these can be done using SEC. So let's say all of the above. Yeah. The absorbance of which amino acid is predominantly detected by the UV detector, right? So the question is which, if I have a detector which is detecting absorbance at 280 nanometers, then which amino acid is predominantly responsible for that? And that is the aromatic amino acid tryptophan. Okay. The other amino acids which contribute to absorbance are tyrosine, phenylalanine, and to some degree cysteine linkages. Okay, but these are very minor. It is mainly tryptophan absorbance which is seen in a UV detector. What can be the possible reasons behind protein molecules not eluting as expected? Column not equilibrated properly? Yes, obviously. Protein has precipitated in the column? Yes. Column is overloaded with the sample? Yes. So all of these can cause the protein molecule to elute in a different manner. Right? What can cause low recovery of protein activity but normal recovery of protein molecules after purification? Right? So the SEC profile is as expected, but when I take my purified protein after SEC, okay, it is not showing the expected activity. So here the options are: protein has been degraded by proteases. This is unlikely in the column, right? Because SEC you're probably doing as a purification step. This is after you've done cell lysis and so on. So most it is unlikely that the protein has been degraded by proteases in this step. Or rather, if the protein has been degraded by proteases, you might also expect the size of the protein to change, right? If this is the case, somehow this has happened. If you do see some degradation, then maybe you'll see that the peak will shift a little bit, right? Or maybe the profile will change. So instead of getting a sharp peak because all the proteins are the same size, you might get some peak like this. Because now my protein is a mixture of different sizes. Column is not equilibrated properly. The column is not equilibrated properly, you will not get the normal SEC profile. Same thing with high flow rate of buffer. Both of these will cause a loss of resolution in the column. Okay, so you will not get the normal SEC profile in this case. So the most reasonable explanation for this is that the protein is unstable or inactive in the buffer that we used for SEC. Which is why it is very important to make sure that it is compatible with your protein. So you should make sure that the pH, the buffer that you use, the salt concentration, if you have to add anything, that should all be compatible with the protein that you're using. Okay, after I've performed SEC, I have eluted the sample in different fractions. So how can they be analyzed? So we can do this mainly by doing SDS pH gel, right? So that would be the obvious way to analyze SEC fractions. I just want to see how much protein is there in each fraction. So what I would expect is, okay, I have some peak like this. Okay, I have collected fractions, right? Maybe after every 0.5 ml of elution, I have collected a fraction. So this is one fraction, this is one fraction, and so on. If I run it in a gel, what I should expect to see is a band corresponding, the thickness of the band should correspond to the amount of protein in each fraction. So maybe here I'll get a very thin band. Here I should get a slightly thicker band. For this, the maximum protein is in this fraction. So I should get the thickest band over here. And I'll get a thinner band here. Thin, thin, thin. And then now there's no protein left in these fractions. So I should not get a band over here. Okay. And by running an SDS page gel from your size exclusion fractions, you can easily identify which fractions actually contain a good amount of your protein, and then you can only use those fractions for your later procedures, right? So here quite clearly I can see that this fraction has the maximum amount of my protein. So I will use that so if I want to do some downstream process, say I want to perform some enzyme assay, or I want to use it for crystallization or something, then I will use this fraction. Okay, I don't have to take this full peak. I just can identify the fraction with the highest amount of protein and use that. Which of the following can be used for cleaning an SSE column? Okay, so SSE columns periodically, because of factors like protein precipitation, other things like that, you can get a buildup of dirt and so on in your SSE column, and that will cause very high pressure in the column. So what we need to do is something called a cleaning procedure. Just give me a second, I'll plug in my laptop and then we'll. Yeah. So when we clean the SSE column, what we do is pass some fairly harsh solutions to denature any proteins or anything else which is stuck inside the column. Okay. So usually we use solutions like 1 molar NaH and 30% isopropanol and pass them to the column so that anything which is stuck inside the column can get denatured and come out. Occasionally, if you think that a lot of protein is stuck in your column, then you can even pass a protease and incubate the column with your protease. That will degrade the protein and then you can elute that from the column. Okay. So, but it's recommended that after every, say, 20 runs, you should clean your size exclusion column. Okay. Yeah. And then I just thought that we can quickly discuss the research problems that Sir had talked about in the course content. So this one is related to oligomeric status of the protein. So PhD student wants to determine the structure of protein X from Mycobacterium tuberculosis. He wants to do X-ray crystallography. Okay. He has cloned the protein in E. coli, set up the crystal, but he's getting precipitate instead of crystals. Now, one thing you should keep in mind for X-ray crystallography is that you should get extremely pure protein in a high concentration. Okay, because you want a crystal that is made up of your protein, and it will not crystallize unless you have a very pure sample. So here he wants to do some troubleshooting and check the quality of the purified protein that he is getting. So he wants to know what can be the molecular weight and what is a native conformation with its oligomeric status. Okay, and obviously both of these we can do through SEC. So I have my known column. Okay, for my known column I will have a known standard graph. Okay, and so what I should do is run protein X in the column. Okay, I'll get some elution volume for protein X. I can then plot it on the graph and get the molecular weight for protein X. Right, so I can estimate what is the molecular weight of protein X by this method. Now another thing I've asked is what is the oligomeric status of the protein. So this molecular weight is corresponding to whatever native status it is in. So if my protein is forming a dimer, okay, then I will get a molecular weight of the dimer itself in SEC. Okay. If my protein is forming a trimer, molecular weight will correspond to the trimer because that is what the native conformation is. But how will I know if it is a dimer or a trimer, right? I'm just getting one value, which is some molecular weight. I'm getting some molecular weight of 60 kDa. How should I know what is the oligomeric status of this protein? So for that, we need to completely break the oligomer and see what the weight of each individual monomer is. And that we can do through SDS page. Right? As we discussed earlier in the course, SDS page is a denaturing electrophoresis technique where, due to SDS and boiling the sample and reducing agents, we can completely destroy the interactions between subunits, get an idea of the true molecular weight of our sample. Okay? So say I run this same sample in SDS page. I have some marker here, and when I'm running this sample, I am getting a band at 20 kDa. Okay, which is telling me that the monomeric mass of this protein is 20 kilodalton. But through SEC, I'm saying that I'm getting that the native mass is 60. So that means that my oligomeric status will be my native mass upon the monomeric mass, right? Which is telling me that this protein is existing as a trimer. Okay? So this way, by combining different techniques, you can tell different things about your proteins. Another way to do this, if you don't want to do SEC, another thing you can do is run a native page. Okay, you can, if you just want to find the oligomeric mass, you can run a native page, right? Where this band should come somewhere here around 60. Right? Since we are not using SDS, the subunit interactions will stay intact, and so we can estimate the approximate native mass of the protein. Now, one thing that the SDS and native page will not be able to tell you is how homogeneous the protein sample is. Okay? Now, from SEC, what you're getting is a chromatogram like this. Okay, I'm getting a very sharp peak. Okay, that is telling me that I have a good homogeneous protein sample. Okay, if instead in SEC, I am getting a peak like this. Okay, that means there are a lot of heterogeneity in my sample. Okay, so maybe my protein is not very stable in my buffer. Okay, maybe it is getting denatured in my purification process. Maybe there is some cleavage or something going on. But this is a very heterogeneous sample, whereas this is a very homogeneous sample, and this will be good for crystallography, where you need a very homogeneous sample. Okay? So if you get something like this, we should try again to optimize our buffer conditions or something so that we can get a nice homogeneous peak, especially if you want it for an application such as crystallography. This is something you can easily tell from the SEC profile. It may not be obvious in the SDS page gel. Okay, in the SDS page gel, I'll just get a band around 20 kDa, I'll be happy. But in the SEC profile, it'll be obvious how heterogeneous or how homogeneous my sample is. Okay. Now saying protein X is present in 3 oligomeric states. Okay, monomer, dimer, and tetramer. Scientists want to study the stability of the protein. So here what they've suggested is using a urea gradient to study what concentration of urea is required to denature the protein. Now, as we discussed, the time taken by the protein to move through the column is dependent on its size, not the molecular weight. So if my protein is in native or completely folded conformation, it will exist as a sphere. But as I keep adding urea, it will start unfolding, right? And as it unfolds and becomes less and less spherical and more elongated, right? Each of these will now act like a bigger molecule in the SSC column, right? So even though the molecular weight of all of these is the same, they will not run at the same spot in an SSC profile. So what will happen is if my native peak was coming here, then as we keep denaturing the protein and the protein starts unfolding, as you can see here, the effective size of the protein is becoming bigger, right? So it will shift towards the left, right? It's becoming bigger, so it will start shifting towards the left, right? So maybe as I add urea, I will start getting SSC profiles like this. And so I can just measure how much shift has taken place, and I can tell you what is the degree of denaturation of the protein, right? Now, urea is one compound that we can use to do this. More commonly, you will see something like guanidinium hydrochloride, or what you can also do is heat the protein. You can heat it to a temperature. So proteins have something called a melting temperature, and at the melting temperature the protein starts to unfold, right? So you can heat the protein and then run it in your SSC column. So here, this is just a comparison of different mutants. Okay, don't worry about what these represent, but I just want you to understand that the blue profile represents the protein as it is, okay, in purified in the native condition and so on. But the red line indicates a sample which you've heated at 43 degrees and then run in SEC. And you can clearly see that when it is unfolded, first of all, the peak has shifted to the left, right? Because the protein is denatured. So here the protein is a glob, but here my protein is denatured, so the peak has shifted leftwards. Okay, and based on the amount of shift, you can get an idea of how stable these proteins are. So all of them have been heated to the same temperature, 43 degrees, but compare this protein, okay? The peak has shifted a lot and it's become really flat, right? So this protein is obviously not stable at 43 degrees. But then compare that to this last protein over here. There's barely any shift in these two, right? There's barely any shift in these two profiles. And the nature of the peak itself is also very similar, which means that this last means that this last protein is stable at 43 degrees and is definitely more stable compared to all of these other proteins, right? So obviously, as we discussed, you can see denaturation by urea, but you can also see denaturation by heat, right? So you can pick a couple of temperature points, maybe 15, 20, 40, 60 degrees, and heat the protein and see how much it has denatured. Another problem. A scientist has isolated a unique protein responsible for induction of apoptosis in the cell. Okay. He suspects the protein might be interacting with DNA. So he wants to design an experiment to study the interaction of DNA with protein. This is something you can do through SSC. SSC may not be the best way to do this, but we can do it. So basically the idea is that we have a protein, okay, and the protein can bind to DNA. Okay? And obviously this molecule is going to behave differently to this molecule in an SSC profile, right? Because this is bigger, it will elute at a different size. So this should elute first, Right? This is my only protein, and my protein DNA complex, which is bigger, is going to shift leftwards in an SEC profile. Okay. Obviously, if you are going to do something like this through SEC, it is necessary to take the appropriate controls and so on. So you should make sure that it is interacting specifically with the DNA. But it can be a very easy way to check if any interaction is taking place. Okay. Um, Earlier in the course, we discussed some other ways to check for this interaction. So we also discussed how we can check for this interaction through something called electrophoretic mobility shift assay, right? Where here we are looking at the shift in the protein profile. So here we are looking at the shift in the DNA profile. So in EMSA, we run basically run the same amount of DNA with increasing amounts of protein to see if there's any shift in the band. Right? So as the DNA binds to the protein, the effective size of the complex becomes bigger, and we see a shift in the band in an agarose shell. Right? So this is something we discussed earlier in the course. So there are many ways to do this. All these binding experiments you can also do through a number of different ways. Okay. So here I'm just listing different ways fluorescence polarization. Okay, these are all just one of many techniques you can do to check for binding. Okay, here's an isotropy. There are many techniques, but of course you can check for binding through SEC as well. I think it's probably one of the easiest ways. You can also do it to check binding between different proteins. Okay, so again, this is my data where what I'm trying to check is I have an antibody. Okay, and I want to check whether it is binding to my protein. Okay, so again, obviously, if I have my protein by itself, it will elute at some volume. But if my antibody is binding to my protein, then this is a larger complex, so it should elute earlier in the SEC profile, right? It should shift to the left. And so here I can see that here is my protein by itself, right? This is my only my protein. And when I'm adding my antibody, right, there is a shift, right? The peak has shifted towards the left because the addition of the antibody has made it into a bigger complex, and that is why the peak has shifted towards the left. So this way I can tell if my protein is binding or not. Okay? Maybe I won't be able to tell the exact affinity. I won't be able to calculate any of the parameters associated with the binding, but I can get a simple yes and no answer. Is it binding or not? Okay? So SSC can be very powerful in this way. One last problem we can solve. Scientist wants to record a CD spectrum, but the protein has been purified by some salting out method, so there's a large amount of ammonium sulfate, and the protein is sensitive to this. So how can we remove ammonium sulfate quickly from the protein? So as we discussed, this is a desalting experiment. We need to use a column with a low exclusion limit. So this would be something like maybe a Cepheurix G25 column, which has very small pores. Extremely small pores. So our protein will just pass all around this, but the ammonium sulfate will have to pass through the pores and will take a longer time to elute. So in the profile of the desalting column, we will expect our protein to come like this, okay, because it is above the exclusion limit of the column. So all our protein will elute together in just after the void volume, and after that, as all the salt passes through the column, we will get a peak for the salt separately. Okay, and this is our protein. So this way we can effectively separate the protein from the salt. Again, SSC with a low exclusion limit. So that is basically what I wanted to discuss today. If you're wondering about when you get when you get a column, how can you decide, or how can you get the parameters for any of the things we discussed today? So when with any column, you'll get an instruction manual like this. Okay, so this is an S200 column. Like we discussed, what is superdex? Superdex is cross-linked agarose and dextran. And here it will tell you about the parameters of the column. So here it is saying matrix composite of cross-linked agarose and dextran, right? Makes sense. It'll tell you the particle size, to tell you the exclusion limit, like we discussed. This is the largest protein that can be separated by the column. We'll tell you the fractionation range. This is for globular proteins. Okay, this is the normal fractionation range. We'll tell you the pH stability range. Okay, so it can what range of buffers you can use with this column. Okay. So on, and it can tell you which temperature range you can use with the column. So if your protein is not very stable, maybe you want to run your SEC at 4 degrees Celsius, and most columns can withstand that. Okay. And most columns will come with their own standard run as part of the as part of the manual. It will also come with a standard run. So here it is telling you, these are the 7 proteins that they've kept as their standard. It gives you an idea of how they are separated, okay, in your column. So you can get a rough idea of what your separation will look like. Okay. So here, say I have some protein which is maybe 150 kDa. Okay, I have some protein which is 150 kDa. And then I can see that the standard of 158 kDa is number 3, which is here. So I will already get an idea that my protein should elute maybe around 12-13 ml when I run it in the column. So if it is eluting at that stage, then I know that my column is working fine. If I'm getting some different elution volume, then maybe I should, that means that something's off with my column or with my protein. So that's something I need to look into. Okay. So this is just a very detailed guide that you will get with any column that you use. Yeah. As we discussed, after you run your column, you will have to do some extensive cleaning sometimes. And that is called a cleaning in place protocol, where we use harsh solvents like 0.5 molar NaOH or 0.5 molar acetic acid. Okay. And after that denatures everything that is stuck in your column. You can then have to wash the column. Okay. You use a lot of water to wash the column properly, and then it is ready to be used. If you want to do more cleaning, you can use things like arginine, or you can directly use a protease. Okay. So the enzymatic treatment, they're saying that you should inject 1 mg per ml pepsin. Okay, just inject it into the column and leave it at 37 degrees. Okay, so then the pepsin can digest any protein which is stuck in the column, and then you can wash out everything else. Okay. So, yeah. So this is what basically you can do with size exclusion chromatography. It's a very powerful technique. I think most most protein labs will do SSC as part of the normal protein purification routine. Okay, I mean we certainly do in our lab, and yeah, yeah. So yeah, so that's basically as much as I had planned for today. If you have any questions related to SSC, we can discuss them. Or if you have any doubts in the yeah, if you have any doubts in the course content that was provided, this we can do it. Or again, if you have any doubts in the middle of the week, please please do make use of this Google forum. Okay, there is a Google group where you can ask things about the video itself. Like here they're asking how to prepare the buffer, right? And someone will answer your question here, right? So it doesn't matter how basic your doubt is. You can always ask here or ask in the problem solving session, right? Even if you feel that there is any issue with the course itself, okay, right. Yeah. So this is the discussion forum where you can ask any question in the middle of the week, okay, and someone will always be able to answer it. Or even if you have any issues with the assignment, you can bring it up over here. So here they're saying that one of the questions in the last assignment was incorrect, right? So this is a way that you can sort of keep in touch with the authorities and the actual people in charge of the course. You can use it to ask questions, and of course you can always ask questions in the problem solving session. Yeah. So yeah, if you have any questions, I can take them now, or and if not, then yeah, I'll wish you a very good evening. And okay, then yeah, then I'll call this session closed. Thank you for your attention, and yeah, if you have any questions, feel free to join in again next week.